I'm very happy to be here and very enthused to talk about the developments in progress that we're making at ImmunogenX. And so I'm going to talk mostly about an enzyme treatment for celiac disease. We have high motivation for why we want to do this. I'm a relative newcomer in the field of celiac disease, having pursued it for about 5 or 6 or 7 years. But the other co-founder of ImmunogenX is Jennifer Seely-Voisner, sitting here, who's our Chief Scientific Officer and is a diagnosed celiac disease patient. And so we have high motivation to try to bring solutions and build a better life for individuals with celiac disease. We were asked to give us our experiences in developing therapeutic solutions, and so one can't really talk about that without talking about the hurdles and the barriers and the challenges that affect not just biopharmaceutical companies like ourselves and a small one at that for us, but all stakeholders that are trying to build a better life for celiac disease patients. And so when we talk about hurdles and challenges, what is it really? We've heard it here. It's about investment, and in this case, a very inadequate amount of investment in the field of celiac disease. And it's not just government funding, and when we've seen that the government really provides a disproportionately small share of funding for celiac disease relative to other GI disorders, and that needs to change, and I think that'll drive many other things, but it's industry as well, and it's also the investment community, the venture capitalists and so forth, who perhaps don't have a full appreciation for the seriousness of the disease, and also don't really understand the market, because there is no market. There are no drugs on the market. There is no market. It's hard for pharma to say how much investment should we put in for a return that we don't know about. Now you take by comparison, irritable bowel syndrome, which I think everyone will say is not as serious a disorder as celiac disease, and there are 5, 6 major drugs on the market that bring in over $4 billion in revenue. And so a pharma company will look at this and say, well, we have a drug candidate that's every bit as good as this one. We're going to bring it out, and we're going to take a good share of this market. We know exactly how much return, we know exactly how much to invest, and that's what is lacking with celiac disease. So that's a hurdle. But I think things are really changing. I mean, just yesterday, the news that Takeda acquired PVP Biologics right after striking a deal with Core Pharmaceuticals. So Takeda, I would say, in my estimation, and maybe everybody else's, is staking a position to be at the forefront in celiac disease market strategy, and Amgen is there too. And so maybe we can look forward to a number of other pharmas following suit. So with that, I'm going to proceed with what I really like to do, which is talk about ImmunogenX. So we're a small company. We have a couple of developments. Our lead development is lattaglutenase, which is an enzyme treatment for breaking down gluten. I'll go into a little bit more detail there. We're in phase 2b trials. We hope to finish those trials and have an end of phase 2 meeting with the FDA leading to phase 3. It is a drug that in previous trials has shown to provide protection of the intestine upon gluten and also symptomatic improvements that are gluten-driven. I won't talk about too much about these others, but I will mention SipCell, a diagnostic, which is a non-invasive monitor of intestinal health, and it uses a drug biomarker. It's actually simvastatin, which is a cholesterol-reducing drug, and it has a remarkable property, which I won't totally go into, but if you ingest it and take 2 blood samples at 2 time points later and measure the levels of simvastatin in the blood, it's a direct measure of the villus health of the small intestine. And it's, we're working with this, and it's an alternative to what is a very invasive biopsy that is expensive and actually not all that accurate, as we've been hearing. They can go up and down. It's a very patchy condition, and so it's not, it might be fine for diagnosing, but it is not for monitoring. It is not accurate enough to be able to detect improvements or degradations depending on how your treatment is going. So we're working on that one. So let me go over we have a fairly strong clinical program. I'm not sure the laser pointer will work over here, but I wanted to first call out the first trial, which was one that preceded us. This was conducted by our predecessors, Alvine, in collaboration with AbbVie. It was a very large trial, 400 completed patients, and it would be what you would call one that delivered mixed results. And the mixed results were because the primary endpoint for intestinal healing was not met. And it was primarily not met, as it was determined later, due to a trial or a Hawthorne effect, which I won't go into, but it's a change in behavior on the patient's part, where all patients, including placebo, improved, and there was no differentiation between the drug and the placebo. And the reasons for that are known. It's not attributed to inactivity of the drug. But secondary endpoints showed some very promising results, in particular for symptom and quality of life improvement, and most specifically for those patients who were persistently seropositive. So we've heard, and I think this ties into the things that we heard earlier, there are patients who don't normalize their serology despite being on a gluten-free diet. Now, some of that is maybe less careful gluten-free diet, but we have quite a bit of evidence to show that some patients are very sensitive. There's maybe a lot of reasons that are not understood for why. There's a whole heterogeneity of responses for patients, and we found that there's a substantial number of patients on a gluten-free diet who remain seropositive, and they were extremely responsive to this drug. So whatever the drug was doing, it was reducing, it was working on gluten. Let me go into the next two trials, are ongoing trials. We call, well, SIPCEL is a trial I talked about, which is a diagnostic, so I'm not going to follow up on that too much unless you have questions. But we have two ongoing trials called Celiac Shield, Solutions for Celiac. One is a gluten challenge trial. The other is a real-world trial. The first one is more than half enrolled. The second one is just beginning. And I was going to bring this up here. Since Francisco made a plug, I'm going to make a plug. So they didn't get into your brochures, but there's a pile of leaflets that describe the two trials that we have ongoing. If you're interested in them, help yourself to the leaflets outside. And we have a third one that's coming online, which is, it's interesting to hear a lot of discussion about T1D in celiac disease. We're conducting a trial at Stanford that will focus on this population base, which really suffer for a lot of reasons, but particularly because the dietary restrictions for both disorders are quite different. So what is there left to eat? So that's going on. What's notable here is that these trials are all funded by NIH grants. We were very fortunate to have been able to win sufficiently large grants to be able to conduct reasonable size trials, and this is really a lifeblood for us. And so this is where NIH has really finally stepped up. And so maybe there's a thaw that will go on, and I think when the government starts to fund this, it will percolate through the whole value chain of investors and pharma and so forth, and we'll get more progress. All right, so let me talk about the drug that we're pursuing. It's called latiglutenase. It's an enzyme treatment. It specifically breaks bonds in the gluten protein to reduce it down to small enough sizes that the fragments, the peptide fragments, are no longer immunogenic. And it basically acts as molecular scissors. And you take this with a meal, and if you eat a meal and you are accidentally glutened, as we all know, we do eat, and we do know we're being contaminated by gluten. And so this is a protection that would prevent that gluten from triggering adverse effects. It's a orally administered drug. It's not injected, doesn't require an IV. And all it takes is emptying a pack of powder into a glass, add water, and you drink it with your meal. And it has flavors. Currently we have 2 flavors. It's appropriately colored for those flavors. It's actually refreshing. Now, I wouldn't call this your drink of choice, but it's not medicine-y. It's pleasing, and you drink it with your meal, and you're protected. And this works in the stomach. The mechanism of action is to break down gluten in the stomach before it reaches its targets. And also, because of that, it's very safe, because it's eliminated digestively. It's not a systemic drug. So let me give you a little bit of clinical data which gives us a lot of confidence that we're going to take this over the goal line and bring it to market to the great benefit of celiac disease patients. This was actually from the so-called, I don't want to say failed trial, but the trial that did not meet its primary endpoint and led to the circumstances of why we acquired this drug. But secondary endpoints for symptom improvement and quality of life were absolutely outstanding. And so this shows percent reduction in symptom severity relative to placebo for 4 gluten-induced symptoms: abdominal pain, bloating, tiredness, constipation. At 6 weeks, you see reductions of 25 to 42%. At week 12, this drug persists. Some drugs sort of lose their potency with time. This persists. At 12 weeks, the improvements are actually greater, ranging from 33 to 56% reduction relative to placebo. So these are quite substantial improvements in this drug. Another thing you look for to prove activity and efficacy is, does it have a dose dependence? And this has a very, very distinct dose dependence. You can see for each of these 4 symptoms a linear increase in the reduction in symptoms with dose. For the higher doses, you see p-values that are below 0.05, and the trend lines, too, are also primarily p-values below 0.05. This is very compelling evidence that this drug works quite effectively to reduce symptoms in this population base. And then this one's a little busy, so I'm not going to walk you through. I'll just highlight it a little bit. This is a similar to what you saw before, only this is aggregating 4 symptoms. This is called the non-stool GI severity scale, and it includes abdominal pain, bloating, tiredness, and nausea. And the results over here show these are the change in baseline at week 12. And you see there's a placebo effect. There are always placebo effects in trials, and that's why you do them. And so, but the improvements are incredibly greater for the 600 milligram to 900 milligram doses. Again, you see the dose dependence that you look for. You see this significant, clinically and statistically significant improvement in symptom reduction, and p-values in all cases there that are below 0.05. So this gives us a lot of confidence we have something that works. And then finally, I'm going to just show this as a safe drug. These are plots of adverse events as a function of dose level. And what you see is there's no dependence on dose. And in fact, I think everybody is saying it looks like it actually gets better with dose. And that sometimes happens because these adverse events, which are very hard for the MD to distinguish from things that are actually gluten-induced. And so in fact, some of these adverse events are gluten-induced and the drug is reducing it. So that's why you see a slight downward trend. But the important thing is there's no upward trend. All right, so to have clinical data that's very compelling gives you a lot of confidence going forward and helps you design what your next trial will be, which will be the phase 3 trial for us. And so that gives us, informs us, as I say, in what we need to do in phase 3. But you also need to have a regulatory environment that's favorable for going forward, and that really means, what does the FDA think of all of this? And fortunately, they are very, very favorable toward the approach that we are using. There was a meeting about 4 years ago, called the Grade 3 meeting. What is that, Dan? Gastroenterology, regulatory endpoints and advanced therapies. Yes, okay. I'll forget that in 15 minutes from now. And one of the conclusions is out of the paper, which I think actually Dan wrote this. This might be his language.
The FDA's language.
Yeah, okay. Yeah.
Funnel it into
You help them with their viewpoint. So basically, symptoms, in particular diarrhea and abdominal pain, will be central patient reporting outcomes for therapies. There's a very high need to improve on symptoms, as long as there's no deterioration of other factors, which will not be the case. And in our Type C meetings with the FDA, they've been very supportive of what we're doing and where we're going, as evidenced by some of the language that's been in the minutes. Clinically meaningful improvements in symptoms represent an unmet need, and that's what we're doing. So we feel good about that, and we think we have a clear road ahead. So I'm going to conclude. I think maybe rather than read through these points, I'll just kind of leave them up there, and thank you for your attention.